the light independent phase, also called the dark reactions. The light independent phase of photosynthesis is basically a biosynthetic phase. This particularly means that something is getting synthesized during this phase. Or in simpler terms, you can say it's an anabolic process. But the question is, how is this a biosynthetic phase? And what is being synthesized during this phase of photosynthesis? The answer to this question is, the dark reactions or the light independent phase of photosynthesis is responsible for synthesizing glucose, which is C6H12 and O6. Now let's see how. The dark reaction utilizes the products of light reaction, that is ATP and NADPH, which we have, in order to produce glucose. And how does it do that? During the dark reactions, the chemical energy of ATP and NADPH, which are produced during the light reactions, they are being utilized to reduce the carbon dioxide into glucose. The light independent reactions occurs through a cycle of reactions known as the Calvin cycle. The Calvin cycle is named after Melvin Calvin, who won a Nobel Prize in 1961 for his work on the details of photosynthesis. Calvin and his colleagues did the work at the University of California, Berkeley. In most plants, carbon fixation occurs through the Calvin cycle. The Calvin cycle is also known as the C3 cycle. And the plants undergoing this cycle specifically are referred to as the C3 plants. Now you must be wondering, what is carbon fixation? Carbon fixation is the process by which inorganic carbon, particularly in the form of carbon dioxide, is converted to organic compounds by living organisms. So, in simple terms, we can define the Calvin cycle as Calvin cycle is a cycle of chemical reactions which are performed by plants in order to fix the carbon from carbon dioxide into glucose. Before proceeding any further, we must be knowing the site of occurrence of the dark reactions. That is, where does the dark reaction actually takes place? As we have studied earlier, the dark reactions takes place within the stroma region of the chloroplast. Now let's talk about the key players of the dark reactions. During the dark reaction, one molecule of carbon dioxide which entered the leaf through the stomata combines with a 5-carbon sugar present within the stroma of the chloroplast called ribulose bisphosphate or RUBP. The enzyme that catalyzes this reaction or that is responsible for combining carbon dioxide with its substrate is known as ribisco, and this is the most abundant enzyme present on the earth. The ribisco stands for ribulose 1,5-bisphosphate carboxylase oxygenase. Now, the C and O at the end of this enzyme's name actually represents its functioning. C for carboxylase, which undergoes carboxylation. And what is carboxylation? Carboxylation means the addition of carbon dioxide with a substrate molecule. Similarly, addition of oxygen to a substrate is known as oxygenation. So, ribisco have the ability to undergo both carboxylation as well as oxygenation. But since the concentration of carbon dioxide within the leaf during photosynthesis or during the daytime is higher as compared to oxygen, so ribisco will always add this carbon dioxide to ribulose bisphosphate or RUBP, and hence the photosynthesis proceeds in an orderly manner, forming ultimately the glucose. Now let's look into the detail of Calvin cycle in a stepwise manner. The entire process of Calvin cycle has 3 important events. These are carbon fixation or carboxylation. And carboxylation of what? Yes, carboxylation of RuBP, because carbon dioxide will be combined with ribulose bisphosphate. Then the second step is reduction. The third is regeneration. Today we will be looking into each of these events in detail. A 5-carbon sugar, as I've mentioned earlier, is present within the stroma of the chloroplast. This sugar is ribulose bisphosphate. It is an isomer of the sugar ribose, which all of you are familiar with. And since it has 2 phosphate groups attached with the 5 carbons, hence it is known as ribulose bisphosphate. Now in the first step, we have to do the carboxylation of this RuBP, ribulose bisphosphate. From the stomata, carbon dioxide enters the leaf, from where, as you all know, it diffuses into the intercellular spaces and then into the mesophyll cells which contain these chloroplasts. The carbon dioxide then reacts, or it combines with RuBP, ribulose bisphosphate, and the reaction, as you all know, is catalyzed by the enzyme ribisco. Now, how many carbons are present in RuBP? Since it's a 5-carbon sugar, it has 5 carbon. And when 1 carbon is further added, when carbon dioxide reacts with it, the resultant compound that forms as a result of this carboxylation must have 6 carbon. So, a 6-carbon compound is formed as a result of the carboxylation of RuBP. This resulting 6-carbon compound is unstable, so it immediately splits into 2 molecules of 3-phosphoglycerate or phosphoglyceric acid. Since 2 molecules of this 3-carbon compound is formed, the total number of carbons present in these 2 molecules will be 6. After the carboxylation, the second step in the Calvin cycle is reduction. But the question is, reduction of what? During this phase, the phosphoglyceric acid or the PGA, the 3-phosphoglycerate, that gets reduced. And you might have learned in chemistry that when an acid is reduced, an aldehyde is formed. Now this reduction occurs in 2 steps. In the first step, ATP from the light reaction is utilized to convert these 2 molecules of 3-phosphoglycerate into 2 molecules of 1,3-bisphosphoglycerate. Now, the PGA or the 3-phosphoglycerate itself had 1 phosphate molecule. But when it got another phosphate from the ATP, it became 1,3-bisphosphoglycerate. That means at this stage it is having 2 phosphate groups, and the ATP after losing 1 phosphate becomes ADP. For each molecule of PGA, 1 ATP is utilized. And since there are 2 molecules of 3-phosphoglycerate or PGA, so 2 ATPs will be utilized in this step. Now, in the next step of the same process, that is reduction, the other product of the light reaction, that is NADPH, that will be utilized. But again the question is, how many molecules of NADPH will be required to reduce 2 molecules of 1,3-bisphosphoglycerate? Yes, 2 molecules of NADPH will be required to reduce the 2 molecules of 1,3-bisphosphoglycerate. That means 1 NADPH to reduce 1 molecule of 1,3-bisphosphoglycerate. Furthermore, 1 inorganic phosphate from each molecule of 1,3-bisphosphoglycerate will also be lost during this step. If you remember, in the previous step, 1 phosphate was added in each molecule, so the resultant molecule that we'll be getting is 2 molecules of glyceraldehyde 3-phosphate, which is a 3-carbon phosphate molecule. And each of this molecule has only 1 phosphate attached to a 3-carbon sugar. Now the 3-phosphoglycerate has been reduced to glyceraldehyde 3-phosphate. After this, what happens?